right? Good morning once again, boys, to all of you. And I'm happy that some of you have done the crossings. Okay. Some of you have put up in the WhatsApp, you have put up your thumbs up, around 6-7 children. I'm happy for them. And someone is putting personally. As I said, instead of putting it personally, put it in the group, so that all of you others who are yet to do, the so-called defaulters, they will also get motivated and start doing it. Initially it was only Sandeep and Kadkol, now the number is increasing. That's good. Okay. Now what we were doing we were discussing the chapter Principles of Inheritance and Variation. And in that, somebody is waiting, Shankar. Okay. Just a minute, I'll just unlock it. Yeah. Okay. So, under this chapter, we were discussing yesterday a very important topic called genetic disorders. And under the genetic disorders, we said there will be two types under the genetic disorders. One is the Mendelian disorder and another is the chromosomal disorders. And under Mendelian disorders, yesterday I think we studied about hemophilia. And we said hemophilia is inability to clot the blood. Clotting of blood will not take place because of absence of particular proteins. And that proteins will not be synthesized in the body of a person if the person has a recessive gene on the X chromosome. And we conveniently, for the sake of memory and easy understanding, we use the alphabet H for hemophilia. And as usual, as the practice given by Mendel, we wrote capital H for dominant gene, recessive, small h for recessive gene, right? And we said that the X chromosome inheritance pattern through various crossings. We have taken various possibilities and did that. And today, as part of it, one more we are going to study, which is called color blindness. As the name suggests, it's a Mendelian disorder, and the person having this disease or disorder will not be able to differentiate various colors that we see. Particularly, I am sure all of us, when we are riding our bike or traveling in a car, we stop at signals, traffic signals. And normally the moment our eye detects red color, we stop our vehicle, because it is a warning for us to stop, not to go. There will be danger. Soon after that, after some seconds, you get the yellow one. That means you are ready to move. And finally, within the seconds, you get the green color, indicating that you can move, you are safe. Now this we are able to distinguish red color, yellow color and green color. And unfortunately, some people cannot distinguish that. So what happens because of that? There can be accidents. Even though red signal is displayed, since person is unable to detect the red color, he moves on, even in the heavy traffic. So invariably, accidents take place. And in our day-to-day life also, it would be very difficult. It is almost, old Hindi movies, black and white, our life will be, since we are unable to detect the colors. Why does this happen? Why a person gets this color blindness? Again, it is unfortunately X-linked recessive disorder. Again, it is X chromosome. Just like hemophilia, color blindness also is, it is sex-linked And in that, there are two allosomes, all of you know, X chromosome and Y chromosome. It is X-linked and it is a recessive gene because of the recessive gene. So it is a disorder due to defect in either red or green cone of eye. Why is that person having this problem can't see the colors, can't detect the colors? You know, in our eye, I think in way back your 9th class, you have studied the structure of eye. In fact, even in 11th class you have studied eye, structure of eye and ear. In that, there are parts in the eye called rods and cones. Remember? So, cones, if there is any disturbance in the cones, we are unable to detect the colors. That means, if cones are disturbed, the structure is disturbed, the number is disturbed, then it results in failure to discriminate between red and green, for that matter even any color. So it is due to mutation in some genes in X chromosome. The other day only we studied. Anything that takes place all of a sudden change in the gene structure, gene locus, chromosomal structure, chromosomal locus, then that results in all of a sudden certain phenotypic characters appearing in an organism. So the phenotype of the organism displays some unwanted characters, undesirable characters. Then we say person is abnormal. Say syndrome. Right. Now, what is the percentage? Look at that. It occurs in 8% of males and only 0.4% in females. And this is due to genes that are linked to X chromosome. Right? Now, he is testing yourself. Can you read the number in this figure? If no, you may have color blindness. I hope all of you are able to read that.
Yes, sir.
Not just by expanding the screen. Yeah, normally. And it is better you check your eyes once in a while. Not only for the color discrimination, see if you have got myopia, hypermetropia, anything like that. Always, you know, prevention is better than cure. And I'm sure in the classroom I was observing you. Some of you who were sitting at the back, last benches, you know, the so-called balcony. Some of you are blinking your eyes whenever I was writing on the board something. When you are shrinking your eyeball, it means you have some discomfort in seeing what I was writing on the board. That means our body is giving signal for us. Come on, check your eyes. If you are shrinking the eyeball and then you are able to see the words that I was writing on the board clearly, it means it is time for you to check for myopia. Okay? And check yourself if there is any bus or vehicle coming, you are not able to spot it at a distance. But when the bus or vehicle comes very close, then you are able to detect. Again, it is time for you to check your eyesight. Of course, Kadkol will be testing it once in 6 months, I am sure. Yes, Kadkol?
I'm having
Yeah, but you need to check. Sometimes, you know, there is something called increasing myopia. The number keeps on increasing, -1, -2 like that. That's increasing myopia. And sometimes it is halted, stalled myopia. That means if your number was 0.5 minus, it remains 0.5. But sometimes your diet also, like you are eating carrots, all green vegetables, then there are chances, and you are continuously using specs or lens, there are chances that the number may come down to minus 0.25. Okay, so you need to check. Whenever you are feeling discomfort, go for the check, and this will be tested even when you clear your UPSC NDA. Particularly if you are a boy who is having passion towards Indian Air Force. Indian Air Force will not accept any defect. So they go for zero error. Okay. Hence, those who are having passion for Indian Air Force, now soon after my class, when you are free, before this weekend, go for your eyesight, check your eyesight, and while you are checking, you check for myopia, hypermetropia, okay, and including this color blindness. That will help you a lot. Okay. Now, what is the various ways that it can come? Like yesterday, all of you have put the various possibilities through checkerboard. Now let us see this. Now first, for your convenience sake, I will read the notes. Normal allele is dominant. Now why C? C for color blindness, right? Recessive allele, that is small c, causes color blindness. The son of a heterozygous woman, I am underlining that word heterozygous, stressing on that, means hetero, different alleles. And what are the different alleles? Capital C, small c. Since it is woman, obviously you are expecting double X, XX chromosome. So she has heterozygous alleles, capital C and small c. And when she is carrying capital C and small c alleles, obviously she is a carrier. Means she is lucky. She will not be having this color blindness. But what happens, Kadkol? When she gets married, produces children, she is going to give a gift to the children. Isn't it? Since she is hiding that allele, that small c, and that is likely to go to the offsprings because of meiosis. So she is a potential carrier. And she has 50% chance of being color blind. Of course, only 50%. A daughter will be colorblind only when her mother is at least a carrier and her father is colorblind. That means you are having this X small c and Y chromosome. Okay. Father's genotype is X small c Y and mother is a carrier. Now let us see various, like yesterday you did know, various possibilities. Let us work out now. Now carrier woman and normal man. Now carrier woman, what is that carrier woman? Just now we said XX, and she will have capital C, small c. Yeah. Normal man, of course, XY. There is no gene there. And what would be the possibility of their children becoming normal or carrier or colorblind? You see, there are three versions. One is the normal child, lucky baby. Second is the carrier, half lucky. And the third one, bad luck, colorblind. So these two couple, carrier woman and normal man, are likely to produce three varieties of children. Normal children, carrier children. Of course, carrier has to be girl only. That's why I said, in this genetic disorders, boys, we are having bad luck. Girls are lucky. Okay? She can become carrier. Sister can become carrier. But she becomes color blindness, okay, possibility is there. But compared to boys, girls are lucky in this way. And then, yeah.
If she is a carrier, sir, it will have any effect
Yeah.
throughout her life?
No, she will not have any. She will be normal. Even phenotype also normal. That is the reason why I said yesterday, in European countries, see, in India, what we do, whenever a boy wants to get married to a girl, traditionally, at least in Karnataka, what they do, they will see Jataka, horoscope. They will count the nakshatra, stars, all that. But in European countries, they are more of scientific. What is that? They analyze the blood. blood sample of the bride, blood sample of the bridegroom. They go for the genetics, gene study. Once they are assured that there are no genetic disorders, hidden ones. See, carrier means what? Hidden one, no. She has that lethal gene. That lethal gene is not doing, fortunately, anything to her. But that lethal gene gets transferred to her children. Isn't it?
Sir, this normal girl who is not will not become a career woman, sir. Then her F1 generation will be all right, no, sir.
Yes, provided she marries normal man. Haji Vikas Yadav.
Means normal man will be having X, Y. Only that is the choice for him, sir.
No, there is. A person can be colorblind, no? If he has got, see, X and Y. If X chromosome has got a lethal gene, color blindness gene,
Yes.
then person becomes colorblind. But he will not tell you, he will not tell the girl that he has got problem. They will manage.
Yes.
That is the reason why many girls are getting cheated, many boys are also getting cheated. And that is the reason why your mother will be more scientific than you and your daddy. When she comes along with you to see the girl, how many questions she will ask? How many tests she will conduct for her? She will ask her to sing, to check her voice. She will ask her to walk, to check if the legs are okay. Isn't it? So your mother is more scientific, right? So we come back to that. A carrier woman marrying a normal man, see, they can produce 3 varieties of children. Possibilities, 3 varieties. Normal children, carrier children, and carrier invariably girl, sister, and colorblind persons, unfortunate. And look at that diagram, you understand. So the carrier woman and normal man. Who is suffering? See the last one, colorblind. Yeah, boy. Isn't it? That is how one has to be careful. Now go to the next possibility. What just now Vikas was telling. A carrier woman unfortunately marries a colorblind man. What are the possibilities? Carrier, normal, colorblind. But look at that. Here, brother and sister both are getting colorblind. If this couple are producing 4 children, 50-50. 50% colorblind, one normal, one carrier. That means the girl becomes colorblind only when her XX is double recessive. Isn't it? Look at that. Contribution from X chromosome of the mother, because she has got that lethal gene in that. And since father is colorblind already, that means he has got already that gene, recessive gene. So X chromosome from father, X chromosome from mother, having the lethal genes are coming to the girl. So unfortunately, the sister becomes colorblind. Whereas, look at the boy who is colorblind. Only one X chromosome. Only one X chromosome. Who is contributing that X chromosome in a male? Mother. So the lethal gene present in the X chromosome of mother is contributed to the son. That's why I said to Kadkol, Tai Kottida Vara. Yes, Sandeep?
Yes, sir.
But of course, I gave a precaution to Sandeep. Mother is mother. It is not intentional. She is also helpless.
Sir,
She is also helpless. If she
a
know,
woman can have,
yeah.
sir,
Tell me.
a woman can be carrier of hemophilia and colorblind.
Yes. Don't think of that. Sandeep, It will be very, very bad now for the family. Possible. Possible. Then you can imagine that family, how much they suffer. There is no restriction that there must be only one genetic disorder in a person or in a family. It all depends on the crosses that we are doing. See, this hybridization crosses, just now we have seen. What is the, like in your mathematics you say no, what is that, permutation and combination? It's all that.
Yes, sir.
Yeah. So, because that is the reason why I gave precaution to Vikas. Be careful when you are going to get married to a girl. Don't see the phenotype. See the genotype also, Vikas. Okay? Genotype is more important than the phenotype. Okay? Right. We come to the next one. Again, I request all of you just draw various possibilities in this color blindness, and again put up thumbs up in our WhatsApp group, and I want all 47 to do it. No excuses. No excuses. Okay? Next, we come to another important one that we have been discussing, sickle cell anemia. And look at that, why this disorder got that disease name, sickle cell. RBC shape is distorted. Now he is showing you normal RBC shape and a distorted one. And that distorted shape is like a sickle. In Kannada you say no, kurpi shape. And being physics students, you know, when it loses its shape, what about the surface area? Okay, so there will be a lot of changes, physiological changes will be there, and this results in low hemoglobin content, low oxygen intake. So consequential things will happen. And again, this is an autosomal, like what we studied above too, hemophilia and color blindness. They are because of X-linked inheritance, whereas sickle cell anemia is autosome linked recessive. It is autosome. How many autosomes are there in us? Yeah?
22 pairs.
22 pairs. So out of that is 44 autosomes. Any autosome has got this recessive trait, recessive gene, that results in RBC losing their shape. Instead of spherical RBC, it becomes sickle shaped RBC. So this is autosomal disorder. So it can be transmitted from parents to offsprings naturally. When both the partners are carriers, see the condition is both the partners are carriers. So it should be heterozygous. So the disease is controlled by a pair of allele called HBA. Why we are writing here HB? Hemoglobin. Good. Hemoglobin A and hemoglobin S. Right? So homozygous, what would be the genotype that causes a person to become sickle cell anemia? It is 3 varieties are there. 3 varieties of genes are there. First one, homozygous dominant. Hemoglobin A, that becomes normal. Second, heterozygous. Hemoglobin A, hemoglobin S, HbS. That becomes carrier. Okay? Carrier. Whereas, who suffers actually with sickle cell anemia? A person who has got hemoglobin S, hemoglobin S. You see, we are writing affected. Now, how does the person get affected? Now, let us see the photos of that. Now, the defect is caused by substitution of glutamic acid. Now, I should tell you that when I say glutamic acid, valine, all this, they are names of amino acids. And all of you know, because amino acids join together and produce a protein. Okay? Amino acids are monomers. Monomers join together to produce polymer. Correct now? So protein is a polymer. Protein is a polymer because it is formed due to addition of monomers called amino acids. So glutamic acid is an amino acid. Valine is an amino acid. Okay? And where is this happening? At the 6th position of beta globin chain of hemoglobin. Now, soon after my class, you need to see, just Google it. What is the structure of hemoglobin? And there is a beta globin chain if you see the structure of the hemoglobin. In that beta chain, 6th position you have to observe. And in 6th position, there is glutamic acid. It will be written as abbreviated as GLU by another amino acid called valine or valine, right? So the shape of the RBC gets changed because of this change, glutamic acid and valine. So this is due to single base substitution at the 6th codon of the beta globin gene from GAG to GUG. Now the same thing, let us see in 6th position. Let me expand that. Yes. Where arrow marks are there, see? There is a change. Glutamine and valine. So glutamic acid or glutamine is out, valine in. And this is a physiological error. And once this valine comes, the person gets affected. The RBC shape gets changed. This is the biochemistry part of it. Yes, histamine. All these are names of amino acids. Okay, leucine. Right. So what you should remember here is that 6th position. And 2 amino acids you must remember. Glutamic acid is substituted by valine. Where is the position? 6th position. Which chain? Beta chain of the hemoglobin. What is hemoglobin? Yes. Hemoglobin is the, now look at that. I am expanding that. Look at that. So it is GAG. Again, I should tell you, I think all of you remember, yesterday I told you that in DNA there will be 3 types of chemical structures, chemical compounds. One is the sugar, I said deoxyribose. Second one is the phosphate molecule. Third one is the nitrogenous bases. And nitrogenous bases, in fact 5 will be there together, DNA and RNA, but in each 4 will be there. And I said A for adenine, it always pairs with T for thymine, and C cytosine pairs with G guanine and vice versa, right? And 3 nitrogenous pairs or 3 nitrogenous compounds will be present together in the form of a triplet code. And I also told you that we will talk about this at length when we talk about biotechnology chapter 11 and 12. At present, you just remember the RBC gets its shape changed because of this. In the beta chain of hemoglobin, at 6th position, instead of glutamic acid, valine has come. Right? This much you remember. Now, let me go to the next one. The same thing is showing you exactly the physiology part of it. See the normal cell, normal hemoglobin, and normal hemoglobin, the disc shaped one. And when it becomes abnormal, see, when it loses its shape, the RBC shape becomes a sickle cell, See the abnormality in the hemoglobin. See that red dots. Can you observe? Yeah. And because of that, what happens? When the shape of the RBC gets into sickle shaped, there is blockage in the arteries. So the blood flow will not be smooth, and that may result in major complications including heart attack. So the mutant hemoglobin molecule undergoes polymerization under low oxygen tension, causing the change in the shape of the RBC from biconcave disc to elongated sickle-like structure. So this is the reason why there is change. So here we are covering both the physiology part of it as well as biochemistry part of it, right? That is the reason why it is getting changed. Now we go to one more that is called phenylketonuria. Normally it is abbreviated as PKU, phenylketonuria. Okay. Now, what happens when the yes, yes, you are right. Only genetically we can correct. There is no cure with some injection or tablet. All these disorders, in fact, until recently they were incurable. But now we are able to cure that through biotechnology. I said rDNA technology, but you should hold on. In India, it is yet to arrive. It is in the still process stage. But in Britain, particularly European countries, there is a lot of advancement in this. So what is phenylketonuria? It's inborn error of metabolism. Again, it is autosomal recessive trait. Just like sickle cell anemia, this is also due to autosomes. And why it happens? Mutation. It is due to sudden change, mutation in the gene that codes for the enzyme phenylalanine hydroxylase. That is, whenever you read a word ending with ASE, you see hydroxylase, you should understand that you are reading about an enzyme. So phenylalanine hydroxylase. What does it do? This enzyme converts an amino acid phenylalanine to tyrosine. Phenylalanine is converted to tyrosine. The affected individual lacks this enzyme. What happens in phenylketonuria? This enzyme will not be there. Which enzyme? Phenylalanine hydroxylase. So if that is not there, who will convert this amino acid phenylalanine to tyrosine? In the absence of that enzyme, phenylalanine stays as such. It won't get into tyrosine. So as a result, what happens? Phenylalanine gets accumulated and changes into phenylpyruvic acid, and it gets into your person's blood. Look at that in the form of, look. So in a normal person, whatever we consume, whenever we consume food, invariably we are taking proteins, isn't it? Whenever we are eating green peas, whenever we are eating vegetables, whenever we are eating fruits, and of course, whenever Vikas Yadav is eating chicken, lot of protein is going there. And that becomes phenylalanine amino acid, and phenylalanine should get converted to tyrosine provided person has got that enzyme phenylalanine hydroxylase, and tyrosine of course melanin and other products. What about in the person that we are talking? Phenylalanine will be staying in the blood like that, and it becomes phenylpyruvic acid. So when phenylpyruvic acid is there, it gets stored. What happens? Look at that. Person will be having these phenotypic characters. That is, phenylpyruvic acid and other derivatives accumulate in brain, resulting in mental retardation. There are also excreted through urine because of poor absorption of kidney. So person becomes mentally retarded. Why person is becoming mentally retarded? Because of the accumulation of that acid. Okay. Now again, one has to be careful about this during the marriage. What is that? Unaffected. Remember, this is autosomal. So unaffected carrier here, since we are talking of autosomes, it can be both. You cannot blame here female, because both of us will be having, both male and female will be having autosomes. So unaffected carrier father, unaffected carrier mother, right? Now, what is the possibility of the children among them? One is unaffected carrier, and 1 out of the 4, means 25% affected. Why affected? Look at that, the deleterious, the lethal gene is coming from both. So double recessive. Okay, and it is autosome. Do not think of X chromosome or Y chromosome. It is autosomal disorder. I think we are getting into the time. As usual, in our WhatsApp group, you put that various possibilities. Let's work out at home and put a thumbs up. Take care. Bye.